Hi everyone, welcome to Module 3. So in the past couple of weeks, we've really been focusing on environmental nutrition as a whole. And starting this week, we're going to get a little bit more specific on issues that may be of concern to you or to your clients, things that your clients might ask you about. So this week, we're going to talk about the impact of pesticides on human health. So our learning objectives are to describe potential impacts of pesticides on human health, identify classes of pesticides that may impact human health, and understand the potential mechanisms involved in the impact of pesticides on human health, including disruption of endocrine function and epigenetics. So what are pesticides? Well, the definition that the National Institute of Health gives for pesticides is shown right here. So pesticide is a substance used to kill, repel, or control certain forms of plant or animal life that are considered to be pests. Pesticides include herbicides for destroying weeds and other unwanted vegetation, insecticides for controlling a wide variety of insects, fungicides used to prevent the growth of molds and mildews, disinfectants for preventing the spread of bacteria, and compounds used to control mice and rats. So as you can see, a pesticide is really a very broad term that encompasses all of these other pest control management methods. Now there are about 20,000 commercial pesticide products that are on the market, and there's over 1 billion pounds of pesticides that are used in the U.S. each year. So this becomes a really important public health issue to understand if these pesticides may have a negative impact on our health. So how are we exposed to pesticides? Well, we can be exposed through a few different methods. We can come in direct contact with the pesticides, so that would be, think about if you spilled something on your hand when you were gardening and applying a pesticide onto your garden, or maybe you touched some mouse poisoning in your house. Or it could be a farm worker who's working on a farm and actually has direct skin exposure to this pesticide. And then we can also ingest pesticides or inhale them. And these are the various methods we would be exposed. So on the right here, I think it's your right also, we have the pesticide cycle. And in this diagram, it's really demonstrating all of the different ways that we might be exposed to pesticides, that they would come in contact with us or we'd ingest or inhale them. So contact is pretty self-explanatory. This would happen for people who are actually dealing directly with pesticide application, like our farm workers or our farmers. And then we have ingestion and inhalation, and this might be come through a couple of different methods that you may not think about. So for ingestion, of course, we have the food that we eat. So pesticides are applied to the food. When we get them, of course, we try to wash them off as well as we can, but there still might be pesticides on the food. So that's kind of an obvious method. But a less obvious method of ingestion would be through our water supply. So here you can see, and this is demonstrated really well in this diagram. So we have our pesticide application, and we can see that, it doesn't stay just on the field. It could run off into a stream. It could seep through the ground, and then it could get into the wells, and then when it becomes incorporated into the groundwater, it can also go back into the stream. So we could ingest it through ingestion of water that is contaminated with pesticides. In addition, we could inhale the pesticides, and that could be, again, through some methods you might not think about. So if the pesticide is applied to the field, then we could have evaporation where it goes into the air. We could have spray drift. So when I sprayed it, it actually just kind of, of course we can't control wind, and it gets blown to a place outside of that direct application location. And we could have the wind erosion, of course. So the tricky part about that is that once it becomes part of the atmosphere, well, of course, we don't have any boundaries in our atmosphere. It can kind of go wherever it wants to go or wherever the wind blows it. But then also it gets incorporated into our precipitation, and then we have this kind of compounded effect where it's running off into the stream and getting into the groundwater again. So pesticides are used primarily in conventional, what we'd call conventional agriculture. And the alternative to conventional agriculture is organic agriculture. So first, before I start on this, I want to give a quick caveat and say when I talk about organic, we're talking about organic practices, but there could be food growers who are following organic practices but are not certified organic. It can be a really expensive certification to get. So just to keep that in mind, I'm kind of talking about organic practices versus certified organic. But the USDA defines organic as a method which produces products using methods that preserve the environment and avoid most synthetic materials, such as pesticides or antibiotics. And then in that same resource, you can go to the link if you want down there, but they just kind of talk about what it takes to grow organic food. So they talk about farmers preserve natural resources and biodiversity, support animal health and welfare, provide access to the outdoors for animals, use only approved materials, no genetically modified ingredients, and keep organic food separate from non-organic food. So in your readings, I kind of focus, so for pesticides, I kind of broke it into two different topics for this module. So for your readings, what it was really our authors were examining was the nutritional components of conventional versus organic agriculture. And so when you're looking at the nutritional components, like the phenolic compounds in others where they do find differences, it's worth recognizing that the organic method does not only include no pesticides, it's also incorporating a lot of these other issues like no genetically modified ingredients. So, and we are going to talk about GMOs next week, but just to keep in mind, organic is more than the pesticides. However, in those articles, they also do talk about pesticide residues and heavy metals, and that is directly applicable to the pesticides. Now, in organic agriculture, you can use specific kinds of pesticides just so that they're not synthetic. So when you see a natural or green pesticide, what should you think? Is this legitimate? Can you trust this? Well, I have a little bit of just a couple minute podcast here from the National Pesticide Information Center that can give just a little insight into that question. Oh, sorry, I didn't press play. Okay, here we
go. Welcome to Pesticides, a podcast series from the National Pesticide Information Center. These are based on common pesticide questions from people just like you.
This is Diana, and I'm here with Umberto to talk about pesticides in terms like safe, natural, green, or non-toxic. Umberto, what do those words mean?
These terms can mean something different to everyone, but they are often used in marketing to convey a sense of safety.
Are these terms accurate when describing pesticides?
Pesticides by their nature are meant to kill or repel a living organism, and therefore have some level of risk. Some of these terms can be misleading when used in relation to pesticides. They can possibly result in carelessness during application. As a result, the US EPA does not allow safety claims such as non-toxic, green, or natural on product labels.
So how can consumers identify pesticide products that are considered low risk?
Consumers can look for products with a signal word of caution on the label, which indicates the product is low in toxicity. There are also pesticide products so low in toxicity they are considered minimum risk and are exempt from federal registration requirements. Consumers may also prefer organic pesticide products that do not contain man-made chemicals or genetically modified organisms. Products labeled organic must comply with specific standards set by the USDA National Organic Program. Finally, antimicrobial products considered to have low impact on the environment may have the Design for the Environment or DFE logo on their label.
That's very helpful. Do you have any other suggestions on how consumers can minimize their risk when controlling pests?
Yes, always read and follow the label directions. You can also consider using an integrated pest management or IPM approach. IPM is a way to minimize pesticide use by incorporating common sense strategies, such as prevention, monitoring, exclusion, and mechanical controls.
Thank you, Umberto.
You're welcome.
If you have questions about
Okay, so that just kind of gave it a quick snippet about how organic farmers may choose to manage pests. So they may use pesticides, but they're not synthetic materials. So as I said, in your readings, the topics were really focusing more on the nutritional composition of the food and the pesticide and heavy metal residues found in the foods of conventional versus organic produce. And for this lecture, I just want to go a little bit deeper. So if we have pesticide residues on the food, what does that mean? It's such a hot topic. You know, pesticides is such a big, scary word. But what does this mean for a healthcare professional who is practicing in an objective, evidence-based manner? So for this lecture, I really combed through the literature and tried to find just what are our general consensus thoughts about the impacts of pesticides on human health. And I'm going to highlight a few of the big topics that I saw that I've seen in the literature. And everything is cited on the bottom, so if you want to dig deeper into any of these topics, please feel free to do so. Okay, so the general consensus is that pesticides may cause acute or delayed health concerns. And I'm going to go into later about what some of our limitations are when we're studying these associations. So on the less severe level, there may be skin or eye irritation or general malaise. So think if somebody came in direct contact with a pesticide, maybe they would have some irritation. Now, more seriously, pesticides, the general consensus is that pesticides may cause negative impacts to the nervous system, and we will talk about why. So some of these effects of the nervous system would include mild cognitive dysfunction, so that would be defined as mood changes or neurobehavioral alterations, so you're just kind of off what your normal neural functioning is. Another would be cognitive and psychomotor dysfunction, minor psychiatric morbidity, depression, and death from mental disorders. And the thing we're going to talk about the most is these neurodegenerative or neurodevelopmental effects. In addition, there's general consensus that pesticides may have a negative impact on reproductive functions. So that would include an increased risk of birth defects, on related note, infertility and impaired fecundability, and then it also may impact altered growth. Now, I was kind of surprised. I really expected to see a lot in the literature about cancer, but there's really a lot of inconsistent evidence, especially due to methodological difficulties. But generally speaking, in review, I think the evidence demonstrates that there is actually potentially lower risk of some cancers, and that includes esophageal, lung, bladder, and colon cancer. But there is generally a higher risk of many other types of cancers. So these cancers include soft tissue sarcoma, Hodgkin's and non-Hodgkin's lymphoma, leukemia, multiple myeloma, stomach, brain, prostate, pancreatic, breast, and ovarian cancer. These have all been associated with varying degrees of consistency to pesticide exposure. And the strongest evidence is for increased risk of hematological malignancies. One health association that I didn't necessarily expect to see, but I have kind of been hearing about, is the impact of pesticides on diabetes and obesity. And we'll talk about a potential mechanism by which the pesticides would have that impact. Okay, so let's talk about where we have the most evidence. And that is with the impact of pesticides on the central nervous system. So many pesticides control pests by acting on their nervous systems. That's the point. They somehow alter nervous system functioning. Either they block signals or they block the blocking of signals so that there's just too much stimulation. So these are really the intended effects on these pests. We want these pests to experience these symptoms so that they die off and they're no longer pests to us. But humans may experience similar impacts. So these pesticides may also have an impact on our central nervous system, which makes sense. So these pests are obviously a lot smaller, so the dosages are very different. It's important to keep in mind. But it's also important to keep in mind that we both have nervous systems and we may have similar impacts on different scales. So one of the diseases that has been studied the most for the impact of pesticides is Alzheimer's disease. So just a little bit of a reminder, Alzheimer's disease is a form of dementia. It accounts for about 60 to 70% of dementia, and it's a progressive, disabling, neurodegenerative disease. Now, moving forward, it's really important to think about the methodology of these studies, and for this topic specifically right now, but also for other topics, we mostly have animal studies and epidemiological studies. So just a reminder, epidemiological studies are where we're looking at entire populations and trying to find associations between the environmental input. That could be anything. Environment doesn't just mean environment. It could be, any lifestyle factor, and disease. So when we look at the animal studies, we do see that some pesticides may cause senile or amyloid plaques and neurofibrillary tangles that we know are defining characteristics of Alzheimer's disease. When we look at epidemiological studies, we do see that there is an increased risk of Alzheimer's disease depending on pesticide exposure. But these are really both difficult, both the pesticide exposure and the Alzheimer's disease are both pretty difficult to study. And we know that there's a lot of variability in the degree of higher risk, depending on the individual, depending on the pesticide, possibly depending on a lot of lifestyle factors that we don't even know to account for. So it seems like in most studies, the general range of increased risk of Alzheimer's disease for pesticide exposure was about 1.5 to about 4.2 odds ratio of developing Alzheimer's disease. So I'm sure we all know what an odds ratio is, but I'm just going to do a review so we're all on the same page. So an odds ratio is saying, you could think of it as how many times the risk someone is having. So if someone had about a 1.5 increased odds ratio, had a 1.5 odds ratio, so we would say those that were not exposed to pesticides have an odds ratio of 1. So that's our reference group. And then we would say, compared to those who did not have pesticide exposure, those who did had a 1.5% odds ratio of developing Alzheimer's disease. So you could say they were 50% more likely to have Alzheimer's disease. And then at the high end, you could say they were 4.2 times more likely, or 420% more likely to develop Alzheimer's disease compared to, 400 times the risk, or 4 times the risk. Does that make sense? 4 times the risk, 400% increase of developing Alzheimer's disease compared to non-exposed individuals. However, again, the results are very inconsistent, so we can't really give a hard number about what exactly the increased risk is. But it's consistently an increased risk. So next, let's look at another degenerative neurological disorder, and that's Parkinson's disease. And this disorder is characterized by the stooping, or postural instability, and the tremor. I'm sure we've all encountered somebody with Parkinson's disease. So the literature suggests that some pesticides may cause mitochondrial dysfunction and Lewy body production that are, again, characteristic of the etiology of Parkinson's disease. So elevated risk exposures were a little bit, had a little bit broader range than Alzheimer's disease. So the odds ratio was 1.4, or 40% higher risk, to 7. So 7 times more likely, or 700 times more, compared to non-exposed individuals. And then the odds ratio increased for those who had had a longer term exposure to pesticides. So think of a farm worker who is exposed to pesticides for these long periods of time. And for those individuals, the odds ratio was almost 6. They were 6 times more likely to develop Parkinson's disease compared to non-exposed individuals. And so I did have a little bit of a list here. Pesticides implicated include, I have 2 specific ones. I'm not sure if I'm saying it right, paraquat, rotenone, and then there's a whole class called organophosphates that are really implicated in these neurological diseases, and we'll talk a little bit more about that moving forward. Oh, I did want to point out one more thing to say that, so for this long-term exposure where we see an increased risk compared to those who may have had less of an exposure, then that's really strong evidence of causality, because we can say there's a dose response, and that's really good evidence to say, okay, if you have a longer exposure or a higher exposure, you should have a higher risk, and we do see that in the literature for this topic. So these are really difficult associations to study for some pretty practical reasons. So why do we have only animal and epidemiological studies? Well, I mean, part of it is an ethical reason. We can't really do a randomized controlled trial where we expose people to pesticides because we know that there is a very real chance that there's going to be negative health consequences. So instead, we need to study this on an animal level to try to find out what the biological mechanisms are, and then on an epidemiological level or population-based level to look for associations or strong associations. But even with those types of studies, it's pretty difficult. So one of the reasons is difficulty in diagnosis. So for both Alzheimer's disease and Parkinson's disease, there's really a lack of specific biomarkers for these diseases. Yes, we do know some things like the Lewy bodies, for instance, but a lot of times we don't have this specific information about what's going on in someone's brain in order to diagnose them. And it's often left up to the expertise of the physician, really, to diagnose these patients with this. So if we don't have these kind of standard ways of diagnosing these diseases, then it's, of course, more difficult to find these associations. There's also a difficulty in assessing the exposure of pesticides. You might not even know what your exposure has been. So if you work on a farm, maybe you're more likely to know what your exposure has been. But if you live near a farm, or if you just have contaminated drinking water, or anything, you may not know which of your food has high pesticide exposure. Maybe someone who washes their vegetables a lot better has less pesticide exposure. It's just really hard to measure. And then another reason that examining these associations is difficult is because we have a lack of important confounding variables. We just don't know what genetic variability will impact these associations. So let me give you an example. During my master's, I took a class on Parkinson's disease. It was extremely interesting to see that even these personality characteristics could put someone at higher risk for having Parkinson's, or whether they drink. I think it was coffee or alcohol or something. You're actually at a lower risk of Parkinson's disease if you partook in those practices. So really, we don't understand all of the genetic variability or lifestyle variability that could moderate the association between pesticides and these neurological disorders. So it makes it difficult to study. Okay, so what pesticides are of particular concern? Well, we have a couple of classes of pesticides that have been implicated in having an impact on neurotoxicity, or of being neurotoxic. And these are called organophosphates and organochlorines. And these, unfortunately, these pesticides are very pervasive in our society. So in one study that took representative biological samples of a representative group from the U.S. population, found organophosphates in 70% of samples and organochlorines in 97% of samples. So as we can see, this is a really big issue where we need to understand exactly what impact these pervasive pesticides are having on our health. So organophosphates are particularly implicated in neurotoxicity, and these really account for about a half of the pesticides that are used in the United States. So these organophosphates have been found to impact synapse formation, axon transmission, cell maturation, and programmed cell death. And they can restrict neurotransmission in the peripheral and central nervous system. So we can see this could really be, something that we would be concerned about. So it really makes sense. I mean, the reason these chemicals are designed to kill insects by inhibiting their neuronal system. So organophosphates specifically kill insects by inhibiting an enzyme that metabolizes the neurotransmitter acetylcholine. And we're going to see, I have an image of that moving forward that shows exactly how that works. But yes, but basically it interferes with nerve transmission and it kills the insect. And we do know that large doses can kill pets and people, but we're still really trying to determine the impact of small doses, especially over long periods of time and especially during periods of brain development. So I believe the general consensus is that these chemical constituents may very well likely have a lot more impact on brains during formative periods of brain development. And they can have lasting effects on learning, attention, and behavior, similarly to what we see with lead. Just a quick little side note, one thing that's been in the news lately too a lot is this issue in Flint, Michigan of contamination of the water with lead. So this is another environmental nutrition issue, where maybe we don't think of our water supply necessarily as nutrition, but of course it is, and we can see that lead contamination still is an issue, especially in our low-income population. So if you're interested in looking that up, I just kind of wanted to make a little plug for that story. Okay, we also see that that prenatal pesticide exposure, so mom was exposed to the pesticide, is associated with increased risk of autism and ADHD in infants and in 8-year-old children. And we also see that research also demonstrates an increase in depression and learning deficits in individuals who are working around pesticides, so may have a much higher exposure than the average person. So this table, or this figure, is taken directly from the article that's cited here, so feel free to go to the article. But it's demonstrating, it's a meta-analysis or review of the associations between autism and estimated exposure of pesticides. And so we have our kind of categories of concern here for pesticides, our organochlorines and our organophosphates, and then here's another pesticide category. So a couple of things I want to point out. First, I want to point out that DDT is listed in this organochlorine, and you may say, why is it even in there? Because it's banned now. We know we aren't using DDT anymore. A couple of things. A, DDT may be being used in other parts of the world where they don't have the same regulations, and as we've seen in the past couple of weeks, we have a global food supply. So I think it's worth thinking about, when we bring food in from other countries, do they have the same safety standards as we have here in the U.S.? It's worth thinking about. And another reason we need to examine DDT is because even though it's banned, it's still certainly it's around. It's persistent. It's what they call one of these persistent organic pollutants. So it sticks around, and we know that it's still present in people's biological samples, including in breast milk. We still see DDT. Another thing I want to point out is just how basically let's just look at what the results were for this review. So again, we have an odds ratio. So this is a pretty typical statistic that we use in epidemiological studies. So an odds ratio of 1 is no difference. Okay, so that's kind of our reference. So that would be our non-exposed individuals, okay? And then anything over 1 is saying you are at an increased risk of autism if you were exposed to this specific pesticide. And anything below 1 would say you're at decreased risk of autism for if you've been exposed to this pesticide. So it's easy to see kind of at a glance where all the evidence lies. So for most pesticides, when there's exposure to the pesticide, there's increased risk of autism. Now, another thing I want to point out is these confidence intervals. So the odds ratio is this dot in the middle, and the confidence interval is the line. So if the confidence interval crosses 1, that's not statistically significant. So even something like this, where we have just a little crossover past 1, this result would not be statistically significant, nor would this one be statistically significant. So we really have like just a couple of them here that are associated with lower risk of autism, and then we have a number of them that are statistically significant for increased risk of autism according to pesticide exposure. So I think another really important concept to think about is, it's one thing when we're ingesting pesticides through our food, and it's another thing when we think about the doses that farm workers are exposed to. So here's a picture here. So in this article from the NIH, and again the link is there, they discuss that 23% of farm workers report a high pesticide exposure event. So where they actually had a really high level of contact, which isn't surprising when we're looking at a photo like this. Of course, you would have high exposure contact. However, having at least one high exposure contact to pesticides, they found resulted in impaired visual scanning and mental processing, regardless of if the person had been diagnosed with pesticide poisoning. This was just self-report of, I have had a high exposure pesticide event. So I'm not going to go into details about the reproductive impact on reproductive function, although earlier in the lecture there was a citation for that, so please feel free to examine that on your own. But I do want to talk about pesticides in diabetes and obesity just because it's such an important topic in nutrition. So we see that I'm also not going to review the cancer studies just because there is such a high degree of variability and inconsistency in the literature. So I will talk about some potential mechanisms by which pesticides might increase risk of cancer, but I'm not going to go into all of the literature. Okay, so let's talk about diabetes and obesity. In vitro studies do demonstrate that the class of organic pesticides, organochlorines, could impair insulin response. Not something you really think about. We will see, when I talk about mechanisms, we're going to talk about how a lot of pesticides are considered to be endocrine disruptors. So if we think about that they're disrupting the endocrine system, it does make sense that they would impact diabetes and obesity. Think about your pancreas. It's a huge endocrine gland that is producing these hormones. So we can see that if we have chemicals that are disrupting our endocrine function, it could result in a higher risk of diabetes and obesity. And indeed, in the literature, we do see an association between consumption of organic chlorine pollutants. So this is indirect consumption. So not I'm working on a farm and I accidentally ingested it. This would be indirect. So like the food I'm eating at the grocery store. And that is associated with higher risk of type 2 diabetes. In general, the literature does epidemiological, animal, and mechanistic data do support a higher risk of type 2 diabetes and obesity for many pesticides. But as we'll see with all of these, it's just there's so many difficulties in methodology to study these associations that we really do not have a good grasp on mechanism, and many gaps in the knowledge remain. So we know that pesticides may alter neurotransmitter and ion channel function, and that could impair pancreatic function and consequently glucose homeostasis, which would put a person at risk for diabetes. For the second article, here's a direct quote from the article. Early life exposure to other subtoxic levels of organophosphates. So when we say subtoxic levels, this is saying these levels have been identified as safe for usage and consumption. So even these subtoxic levels could result in prediabetes, abnormalities in lipid metabolism, and promotion of obesity. So these are some pretty big risks that we're talking about when we're talking about pesticide exposure, especially because it is such a big exposure to so many people. And it's kind of goes beyond our choice of what foods we're choosing because it's also in the environment and in the water. And yet, these topics are so difficult to study, so we really don't have a very good grasp on mechanisms. There's a lot of hypotheses, and there's substantiated hypotheses, but really we still have a lot to learn about how these impacts are also actually being carried out on a biochemical level. And we have a few ideas of how this is working. So the first thing would be that these pesticides increase reactive oxidant species production and also deregulation of antioxidant enzymes. So it's easy to understand that if we have these increases in ROSs and decreases in our antioxidant enzymes, this is going to eventually lead to inflammation. And when we have inflammation in our nervous system, this could lead to neuronal loss. So that's one potential mechanism. Now the next two mechanisms, epigenetics and endocrine disruptors, I'm going to go into in more detail. And just keep in mind that these are not all distinctive mechanisms, but there's actually some overlap in these mechanisms. So the first thing I'm going to talk about is epigenetics. And for those of you who had me last semester, this is a little bit of a review, but actually very applicable to the topic at hand. So for those of you who haven't had exposure to epigenetics, this is a field of study that involves genetic control. Sorry, this is just, epigenetics is a field of study, but it's actually also what's happening on a microscopic level. And what it is it involves genetic control by factors other than the individual's DNA sequence. So the concept of epigenetics is that, maybe we would not have a change in our DNA sequence, but when we have different environmental exposures, it can either turn on or off the expression of our genes. So our genes themselves may not change, but whether they are expressed or not and translated into proteins, that could be altered by environmental exposures, and that includes pesticide exposure. And the literature demonstrates that environmental exposures during formative periods, such as the prenatal period or early postnatal period, could increase risk for neurodegenerative disorders later in life. And epigenetics may be one of the mechanisms by which pesticides have noxious effects. So I'm going to go to a YouTube video here. It's not much about pesticides, but it's more about epigenetics to introduce to give you a better understanding of how this would actually work.
Imagine coming into the world with a person so like yourself that for a time you don't understand mirrors.
As a child, when I looked in the mirror, I'd say, "That's my sister." And my mother will say, "No, that's your reflection."
It's hardly surprising, because you both come from the same egg. You have precisely the same genes. And you're literally clones, better known as identical twins. And yet, it's not uncommon for a twin like Anna Marie to get diagnosed with cancer, while the other, like Clotilde, doesn't. But how can two people so alike be so unalike? Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Jerdal. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones?
That's correct.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time, causing obesity. So what accounts for the thin mouse? A tiny chemical tag of carbon and hydrogen, called a methyl group, has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.
In fact, it's the epigenome that tells our cells what sort of cells they should be. Skin, hair, heart. You see, all these cells have the same genes, but their epigenomes silence the unneeded ones to make cells different from one another. Epigenetic instructions pass on as cells divide, but they're not necessarily permanent. Researchers think they can change. And that brings us to the reason why we're showing you twins. In 2005, they participated in a groundbreaking study in Madrid. Its aim? To show just how identical epigenetically they are or aren't.
Our genes are just part of the story. Something has to regulate these genes. And part of the explanation is epigenetics.
Estella wanted to see if the twins' epigenomes might account for their differences. To find out, he and his team collected cells from 40 pairs of identical twins, aged 3 to 74. Then began the laborious process of dissolving the cells until all that was left were wispy strands of DNA, the master molecule that contains our genes. Next, researchers amplified fragments of the DNA until the genes themselves became detectable. Those that had been turned off epigenetically appear as dark pink bands on the gel. Now, the genes from a pair of twins are cut out and overlapped. The results are far from subtle, especially when you compare the epigenomes of two sets of twins that differ in age. Here on the left is the overlapped DNA of 6-year-old Javier and Carlos. The yellow indicates where their gene expression is identical. On the right is the DNA of 66-year-old Anna Marie and Clotilde. In contrast to the younger twins, hardly any yellow shines through. Their epigenomes have changed dramatically. The study suggests that as twins age, epigenetic differences accumulate, especially when their lifestyles differ.
One of the main findings of research is that epigenomes can change in function of what we eat, of what we smoke, of what we drink. And this is one of the key differences between epigenetics and genetics.
Okay, gotta love that Neil deGrasse Tyson. So that's just kind of an introduction to epigenetics. Now, they described in the in the clip how epigenetics works, but I'm just going to do it in a little bit more detail. So epigenetics can work, or our environment can impact what genes are expressed based on 3 different mechanisms. So the most studied is DNA methylation. The next is histone modifications. And then we also have RNA silencing, or these microRNA RNAs. Okay, so for DNA methylation, when DNA is methylated, basically a methyl group is added onto a DNA strand at a really specific location. So it's between the C and the G bases and linked by a phosphate. So this is called the CPG site. And generally when a gene is methylated, it stops the expression of this gene. So you can see this in this graphic here. We have methylation at this very specific site, and it's saying, okay, we are not going to express this gene. Whereas if the site is not methylated, then the gene can be expressed. So hypermethylation is associated with decreased gene expression, whereas hypomethylation of these non-coding regions, or these regions that shouldn't be expressed, that's linked with chromosome instability. So really, we kind of want these methyl groups exactly where they should be so that we have correct gene expression. And if they're not in the right places, we can have hyper or hypomethylation, both with consequences on genetic expression. The next one is histone acetylation. So just as a reminder, a histone is a family of proteins that is associated with DNA in the nucleus and help condense the DNA into chromatin. So let's go back up. So here we have the histones, and here we have the DNA that's wrapped around these proteins, and basically it keeps it nice and condensed so that it can fit in the nucleus of the cell. So it doesn't appear, we know that DNA is not in linear strands when it's in the nucleus. Instead, it's condensed and it's wrapped around these histones inside the nucleus. Now, when we add an acetyl group on to the histone, basically the lysine on the histones removes a positive charge, and this reduces the affinity between the histone and the DNA. So that when an acetyl group is added, it really loosens the chromatin around the histone and lends itself to replication and transcription. Whereas if we have a histone that doesn't have the acetyl group on it, then it's really kind of still really tight around, the DNA is still really tight around the histone, and it restricts access to various enzymes so that it won't be expressed. So in this way, histone modifications can regulate gene expression, chromatin modeling, cell survival, and cell death. And it's really this histone structure that could mediate pesticide toxicity. And here's a little graphic here too. Whoops. Just showing that if it has the acetyl group on it, we're kind of loosening up this, and we can have transcription of the gene. Whereas if we don't have the acetyl group, we have the tightness of the chromatin around the histone, and this will block expression of the gene. And then finally, I'm not going to go into too much about this, but we have this these microRNA that can mediate mRNA expression. And these microRNA are just these little single strands of RNA that are just around 20, 25 nucleotides. And these kind of pair with the mRNA in order to regulate the mRNA's stability and how it's going to be expressed or transcribed into proteins. Now, there's a whole field of nutrition epigenetics, and this is really examining especially nutritional environment and how that impacts, nutrition environment during formative periods and how that may express later disease risk. And this is really a developing area of study. And so the premise is that early suboptimal nutrition could have health consequences later in life. And we do see in the literature that this is the case. So nutrient exposure during formative periods can impact how genes are expressed and can impact risk of cancer, aging, obesity, type 2 diabetes, inflammation, and neurocognitive disorders. So if you look at this list, what comes to mind? What comes to mind for me is that there's a lot of overlap with the chronic diseases that are implicated in pesticide exposure. So there could be a lot of overlap, whereas in nutritional epigenetics, we may be more focused on was there enough nutrition or overnutrition during these periods, calories, and exactly what we've talked about in traditional nutrition. However, really, I think it's worth considering whether it's really the pesticide exposure on these foods that could be causing these epigenetic changes, or a combination. It's just worth thinking about. Okay, so what does the literature have to say about pesticides and epigenetics after that very long intro? Well, all of these are almost direct quotes from the articles that are cited below, because I think the authors just put it the best. So we know that several epigenetic mechanisms, including DNA methylation, histone modification, and microRNA expression, can change genome function under environmental influence, such as environmental pollutants. So we know that environmental exposures to these persistent pollutants and endocrine disrupting chemicals, which I'm going to talk about next, has been shown to modulate epigenetic markers. Early life exposure to endocrine disrupting chemicals, again, we'll talk about that next, do have lifelong effects on neuroendocrine gene expression and on reproductive dysfunction. Now, this middle point, I think, is a really interesting point. So, like I said, I was kind of surprised at the inconsistent results of pesticides on cancer. And what's really interesting is that when we think about the safety of these pesticides, they all have, the ones that are in use have nearly all been approved by the EPA as not having direct toxicity. So it's interesting because these pesticides don't increase cancer risk via directly genotoxic mechanism necessarily. Instead, they could operate through epigenetic changes. So they could be really altering how our genes are expressed that may impact later disease risk. We do see in animal studies and in vitro studies that alteration of DNA methylation patterns have been increasingly found in tumors, especially those of hematological malignancies. Again, this should stick out for you because we know that this is kind of the strongest evidence we have for pesticide and cancer risk. And then that there are other possible mechanisms involved in tumorigenesis, including this reactive oxygen species generation, endocrine disruption, and DNA damages. So really the pesticides could impact many different mechanisms in order to have the increased risk that we note. Okay, and I'm sorry this is pretty tiny, but you can go to this, hopefully you can see it okay, and then you also can go to the source if you'd like to. But this is a really nice table that's demonstrating the evidence that we have of epigenetic modifications induced by pesticides. So here we have our class of pesticide. Here we have the exact component of the pesticide that may be detrimental, what epigenetic changes it's making, what type of organism this has been studied in. So we can see we do have some human studies, but really important to know, what do we mostly see here? Mostly in vitro studies and animal studies. So, So that needs to be considered when we're talking about applicability of results. And then it also, this table also demonstrates the tissue which is impacted. Okay, so let's take a little bit of a movement from epigenetics to endocrine disruptors. And again, these aren't completely separate mechanisms. They are related. So endocrine disruptors, or endocrine disrupting chemicals, sorry, I see a little typo here. This is supposed to be EDCs, endocrine disrupting chemicals. Okay, so these are synthetic chemicals that resemble natural hormones. And then because they resemble natural hormones, either mimic or antagonize our hormones that we would naturally have endogenously, and therefore cause these epigenetic changes. So we know that some pesticides do interfere with hormone receptors, hormone synthesis, or hormone conversion, and we call these the endocrine disrupting chemicals. And it does seem pretty clear that these endocrine disrupting chemicals do have more of an impact on a developing organism, such as a prenatal fetus or postnatal infant, than they would compared to adults. And this makes a lot of sense because this is a period where we are really dictating what our gene expression will be during these formative periods. So here's a nice little graphic. It's not as nice as I wish it were, but from the NIH. And so this is showing this first one is showing everything going right. So we have our normal hormone in our cell. The hormone lands on the hormone receptor and then instigates this appropriate chemical response. So here we have that an endocrine disrupting chemical could be a hormone mimic, okay? So in that case, it would land on the hormone receptor and cause the same cellular response. However, not really demonstrated great in this picture, if we have, our bodies produce a certain amount of hormones. So if we have another compound that is acting as a hormone, well, then we could have kind of over response of the, exacerbation of the hormone response that our body was intending to have. Also, we could have antagonism from these endocrine disrupting chemicals. So again, I wish it was a different color, but this is showing that we would have our hormone blocker or our antagonist, which would antagonize our actual hormone from landing on the receptor and having the intended response that we wanted it to have. So it would actually block hormone action. So you can see it can either make the stimulation, exacerbate the stimulation, or block the stimulation. And here's a really nice figure about the impact of the class organophosphates, which are endocrine disrupting chemicals. Look, I did it again. Jeez Louise. Ah! I'll post it correctly. Don't worry. Okay, so here we go. I'm sorry. Just a second. Okay, so here we're showing that organophosphates could impact acetylcholine signaling. So we all know that acetylcholine is a neurotransmitter. So in this green box, it's showing how acetylcholine is supposed to work. We have acetylcholine here. We have our acetylcholine receptors here. The acetylcholine lands on the receptor and has our intended chemical or nervous system response. Perfect. That's how it's supposed to work. Here, we have a normal system. So we have acetylcholine esterase, which stops the signaling process. So basically, our body is able to say, okay, in this case, I wanted the acetylcholine to have this intended nervous system action, but now I'm done with that signaling process. So then my body is going to produce this acetylcholine esterase, represented here by a star, and that's going to block acetylcholine from landing on its receptor and then having signal transmission. Now in this last red box, the figure is demonstrating how these processes are altered by the pesticide class of organophosphates. So organophosphates inhibit acetylcholine esterase. And remember, acetylcholine esterase stops the chemical transmission of acetylcholine. So here we say, okay, so I have my acetylcholine, and I have my acetylcholine esterase because my body's saying, stop with the signal, we're done signaling. But then my organophosphate pesticide comes in, and it actually blocks the action of acetylcholine esterase, so that really my acetylcholine keeps landing on these receptors, and then I have more signal transmission than intended. So think about something like Parkinson's, where people are not able to control their motor function. This really makes sense as a mechanism. Okay, so this figure on the left here demonstrates how exposure to the endocrine disrupting chemicals could impact later disease risk. So it's highlighting that the early life exposures to these chemicals could cause functional changes at a cellular level that cause physiological changes and disease risk. So here we have our cell, okay, and then we have development at this formative period of the organism. And so here we have a critical window to susceptibility. So this may be why we have a greater impact. I mean, other than dosage, of course, is a consideration as well, but also a developing organism may have this kind of critical window of susceptibility. And if they are exposed to these endocrine disrupting chemicals during that stage, then that could alter what genes are expressed on these somatic cells. And that gene expression could follow that organism for the rest of their life and really could increase susceptibility to disease later in life, well after the exposure even occurred. Now, this figure is demonstrating a correlation, so just keep that in mind. I'm not trying to suggest causation, but it is really interesting because it's demonstrating the correlation between synthetic chemical production in the U.S. and prevalence of type 2 diabetes. So as we discussed, endocrine disruption really would make sense as etiology, or at least contributing factor to the etiology of diabetes. And we do see that indeed we kind of have an uncanny association for the increase in synthetic chemical production and the risk of diabetes. Okay, and this last slide is from an article that was examining endocrine disrupting chemicals. So as I've already said, examples of ECDs include these organophosphates and organochlorines, as well as a class called carbates, and DDT, which is banned but still found in our tissues and biological samples. And we do know that these endocrine disruptors can result also in menstrual irregularity and changes to fertility. And again, that does make sense if we're thinking that these endocrine disruptors, look, I did it again. I will fix this before I post it, but just so you know, endocrine disrupting chemicals, EDCs. But anyway, this does make sense because if these are mimicking hormones, it's really easy to see why they would have an impact on the menstrual cycle and consequently in fertility. And here are some specific pesticides that are recognized as endocrine disrupting chemicals. So we kind of have our classes here and then some specifics here. And here are the organizations that recognize these pesticides as endocrine disruptors. Okay, so that is all that I have for you today. Hopefully I gave you at least a decent overview through the readings and through the lecture of the association between pesticide application and how that may impact our the nutritional composition of food, but particularly how that impacts the pesticide exposure of food, and then the impacts that may have on human health moving forward. So again, this is a huge topic. Please feel free to pursue any of the references that are noted in here if you're interested in a specific topic. All right. I hope everybody has a great week and enjoy the rest of your day.